Hello students and welcome back to the chapter, and in this module we shall be having a look at replication of bacteriophages. We have already studied what bacteriophages are, so now let us study how they replicate. So bacteriophages replicate by the lytic cycle, and we shall study this lytic cycle in a stepwise manner. So here I have a bacterium which shows me its nucleoid, and here I have a bacteriophage. Now this bacteriophage is going to attach onto the surface of the bacterium, that is the cell membrane of the bacterium. So the first step in lytic cycle is attachment. And attachment of what? Attachment of bacteriophage onto the bacterial cell. And this is dependent on random encounters with the correct receptors. And once attached, this bacteriophage cannot move because the attachment is very specific. So now let us move ahead and study the second step, which is called as penetration. So here can you see that the viral DNA has been inserted or penetrated into the bacterial cell? How is this achieved? This is achieved by the contraction of the tail fibers. The tail fibers contract and then they bring the base plate closer to the surface of the bacterium, and hence the viral DNA is injected into the bacterial cell. And now can you see the empty capsid or the empty bacteriophage body outside? So this empty capsid is termed as ghost. So this is the second step of penetration. Now let us move ahead and take a look at the next step. Here we have synthesis of proteins and nucleic acids. The host's DNA replication synthesis and protein synthesis is disrupted, and it is compelled to make the viral DNA and proteins. So that is how the synthesis of proteins and nucleic acid is carried out. The next step we have packaging of DNA. Can you see that the DNA is packed into the capsids, and also the other parts of the bacteriophage have been constructed, the tail fibers and the base plates. So this was packaging of DNA. So now let us move ahead and take a look at the next step, which is virion assembly. The new viruses are termed as virions, and can you see that they have been assembled? They resemble the original bacteriophage structure. So the DNA is packed into the capsid and then it is assembled with the tail fibers and base plates. So this is virion assembly, and this entire process, that is, these are 4 steps, take 15 minutes, 15 minutes for completion. And the final step we have here is the release of virions. So these newly formed virions are released from the bacterium, and it is achieved via the lysis of the bacterial cell, and it is catalyzed by an enzyme called as endolysin. And now these newly released virions can go and infect new bacterial cells. So that is the lytic cycle. And now let us take a look at the lytic cycle on whole. So here we have attachment, then we have penetration, then we have synthesis of proteins and nucleic acids, then we have packaging of DNA, then we have virion assembly, and finally we have the release of virions. So that is the lytic cycle, my students. Now let us move ahead and take a look at this picture. Here you see a bacterial lawn that is grown on agar surface, and the holes or the empty spaces that you can see there are termed as plaques of lysed bacterial cells. But what use are they? Now the cloned viral DNA or the cloned DNA can be recovered from these plaques. So that is how the replication cycle of the bacteriophages, that is the lytic cycle, helps us to obtain multiple copies of DNA by cloning. So in this module we studied replication of bacteriophages, and in the next module we shall be studying restriction fragments and restriction endonucleases. So till then, study well, take care, and bye-bye.